Hello everyone, my name is Ibrahim. I'm a PhD student, actually graduated last week in the lab of STEM Arts. Today I will talk about how we use deep learning models to design synthetic enhancers. Enhancers are one of the core elements of gene regulatory networks, where transcription factors, they recognize and bind short patterns on the DNA sequence, and their combinatorial and differential expression of these transcription factors regulates cell type specific gene expression. And recently, we studied the code of enhancers by training deep learning models on the DNA sequence in two different systems, melanoma and Drosophila brain. And in this study, today I will present by using the models that we trained before we use them to design synthetic enhancers. And the search space, even for a 500 base pairs sequence, it's enormous. So for this reason, we take three different approaches, which are nucleotide by nucleotide, motif by motif, and the whole sequence at once. To start with the melanoma part, I will give a brief introduction. So what we did to get the candidate enhancer sequences, we performed a TAC-seq on 17 patient-derived human melanoma cell lines, and we clustered co-accessible regions by using cystopic, and then we trained the deep learning model on the DNA sequence, where the model takes 500 base pair DNA sequence as input, and it tries to predict in which cell type that given sequence will be accessible. And to give an example how it works, here you see the IRF4 locus. The enhancer is accessible in the fourth intron only in the MEL subtype, but not in the MES subtype of melanoma. And if we score with the model, looking at the MEL subtype, the model says the enhancer is here. Then we ask model by using the tools like Deep Explainer or Deep Lift, how can you know that there is an enhancer? And it shows us the most important nucleotides that are used by the model to make a correct prediction. And if you zoom into these most important nucleotides, here you can see we can identify different transcription factor binding sites, activators, and repressors. This is one way to understand what model sees on the sequence, and the other way is like doing perturbation assays. Here we mutate each nucleotide to other three nucleotides one by one and calculate delta prediction score. It's called saturation mutagenesis, and we do in silico. And for this IR4 enhancers, actually, we were lucky because the Kircher et al., they did the same experiment in vitro. And as you can see, like our model prediction correlates quite well with the in vitro assays. And actually from this picture, we learned a lot. So if you want to decrease the activity of an enhancer, you can decrease the activator binding sites, you can destroy them, or actually you can create repressor binding sites that will decrease the activity as well. Here you can see these individual mutations are actually each of them, they are creating ZEP2 binding site, and they decrease the activity of the enhancer. And to increase the activity of the enhancer, you can destroy repressors, you can create new activated binding sites, but also you can make the already existing binding sites better to increase their affinity, like this mutation creates a better MITF binding site, or this one a better SOX10 site. So we saw that, okay, on a given enhancer, we can go to both directions, increase the activity or decrease the activity of the enhancer. Inspired from this, we took a sequence from another species, dog here, which was accessible and active in dog. And here you can see the model prediction and the luciferase activity. And we performed only these two mutations. One of them creates better SOX10 binding site, the other one creates repressor binding site, at the same time creates activator binding site. And with these two mutations, we were able to increase the activity of the enhancer tenfold. And another application, this time we took two orthologous enhancers, which dog part is accessible and active, but not the human one. And here you see the prediction score versus the activity for the human and dog. And following, again, the saturation mutagenesis, we performed these for mutations, and we were able to rescue the activity of an enhancer, probably that was lost during evolution. Okay, we can enhance an enhancer. We can rescue an enhancer that was lost. So we asked, can we create an enhancer from scratch? Here, the strategy is quite straightforward. We start with a random sequence, numpy random, and then we do saturation mutagenesis, and then we choose the best mutation, and then we apply the mutation, and we do this procedure iteratively. Here you can see how the prediction score is changing with the number of mutations. And we selected 10 of them randomly to see if they are really functioning like an enhancer. And we test them in vitro, again, with the luciferase assays. As you can see with the reds, these are the genomic ones. Our design sequences distributed quite well in between the genomic enhancer activity levels. Here you see an example. This is the initial random sequence. There are repressor binding sites and no activators, and it's by chance the repressor binding site here. And after 15 mutation, activator binding sites are generated and the repressor binding sites are destroyed. And to see what's happening in each step, here you can see like the random sequence, after 3 mutation, it created this SOX10 binding site here. And also we tested these individual steps to see how the activity of the sequence is changing in vitro. And as you can see, for example, the initial SOX10 alone doesn't do much, but after there is the other cofactors, adding a new SOX10 is actually causing a high jump. And we were able to track what each mutation does and which transcription factor binding sites they create and how it affects the activity of the enhancer. And focusing on again on this same sequence, random 1, and after 15 mutation, this time actually, okay, repressors, we saw that repressors are actually blocking the activity of the enhancer. To test this on this evolved sequence without touching the activator binding sites that was created from random sequence, we introduced these single mutations that each time it creates a ZEP2 binding site. And then here you can see the prediction score after adding these repressor binding sites. The model says it's not an enhancer anymore. And if we test this in vitro, after 15 mutations, if we add the repressor binding sites without touching the activator binding sites, now the activity of the enhancer is gone. And also we wanted to check, okay, like now we are adding 7, 8 repressor binding sites, then we wanted to see if we add them one by one, cumulatively, as you can see, the more repressor binding site we add, the activity of the enhancer decreased. And if we add 7 or 8, now the activity was lost completely. And also what we tested is, it's okay, we are testing these enhancers with the separase assay, we see the activity of them, and but we wanted to see how the accessibility is changing also. We integrated these sequences into the genome of our cell lines. And here you can see the random sequence, it's not accessible. After 15 mutation, now the region became accessible. And if we add repressors on top of this accessible region, now the accessibility is almost gone. And so far, what I showed is actually was on human cell lines, but we wanted to do this in a more complex system where there are many different neuronal or glial subtypes, which is Drosophila brain. So to give a very, very brief introduction to Nature paper, we performed single-cell task-seq on developing fly brain. We further clustered adult glial canyon cell and T neurons further. And then we again, similar to melanoma, we performed cystotopic analysis to identify cell type specific accessible regions. And then we trained the models on these DNA sequences. And by using the deep explainer, deep lift, and the TF-MODI score, we were able to identify the main transcription factors for canyon cells, one-cut islets, and MEF2 as activator. And also we were able to identify cell type specific repressor factors, where when the transcription factor is expressed, it is actually contributing negatively for the subtype it is expressed. And even though those activator binding sites are present, when the repressor transcription factor is expressed, it is repressed in the cell type that is expressed, and also if you look at the target gene, it's not expressed either. I would like to give an example, it will be more clear. Here you see the SNPF gene, its enhancer is here, and it's accessible in 2 out of 3 canyon cell. And if you look at the single cell RNA-seq to see the gene expression of the SNPF gene, it is expressed on those 2 subtypes. And if we create an enhancer reporter assay and test in vivo with the GFP, you see that it is okay. And canyon cell enhancer, it's expected at its location. And if you score this region with our deep learning model, okay, it says also this, it is 2 out of these 3 canyon cell subtypes. And then we ask model, how, and why do you know, how do you know this? And then again, it shows us the most important transcription factor binding sites, namely SRMF2 iris 1 cut here for the canyon cells. And the reason why in alpha prime, beta prime, the region is not accessible and the target gene is not expressed is because of these repressor binding sites. They like actually you see the same DNA sequence on these 3 rows, but through different glasses through the model. These are the contribution scores for alpha beta canyon cells, and these are contribution scores for the alpha prime beta prime canyon cell neuron at the end of the model. And then it means that the binding sites that are bound by this repressor transcription factor exist in all cases, but the transcription factor is expressed only in this case. So that's why the region is not accessible anymore and the gene is not expressed. And focusing on these activator binding sites, with the help of saturation mutagenesis, we destroyed MEF2 binding site with these two mutation or ILIS binding site with these two mutation, and each case it destroyed the enhancer activity, which shows that the binding sites we actually identified are correct ones, real ones. And okay, this similar to melanoma, then we asked, can we create an enhancer from scratch? And here, number of mutations versus prediction score you see. And then after 10 mutation, if you look at the distance between the farthest mutation, it was like around 200 base pair, which corresponds to size of a nucleosome, which means it's actually model is trying to create an enhancer by using just one nucleosome size. And you can see an example here is the random sequence. By chance, there are a short repressor binding sites, and after 10 mutations, repressor binding sites are destroyed, and activator binding sites for the Kenyan cells are generated. And if we tested the 13 of them that are evolved from random sequences, and here you can see 10 very nice positive ones that our success rate was quite high. And also, like we went further, okay, like starting from a random sequence is not active, and we make a Kenyan cell enhancer. Then we want to do actually starting from the same random sequence, can we create another enhancer that is targeted for the another cell type? Here is perineuronal glia. Actually, these are two same random sequences, but through the eye of Kenyan cell or perineuronal glia. And the model is actually selecting different mutations on the, if you direct to KC or PNG, and at the end, either we create perineuronal glia enhancer or Kenyan cell enhancer, starting from the same random sequence. And another thing we tried is actually enhancers. They are not always cell type specific. Some of them, they can be accessible and active in multiple cell types. And if you can create an enhancer from scratch, we thought we can maybe create an enhancer inside another enhancer, two overlapping codes. Here is an optic lobe specific enhancer. It's accessible in T2, T4, and T5 neurons, and the model predicts correctly. Here, this time we choose the mutations that doesn't affect the T neuron prediction score, but increasing the Kenyan cell prediction score. And after 13 mutation, we were able to spatially augment the activity of this enhancer. And the other way around, we can find such regions on the genome. You can see gamma Kenyan cell prediction score versus T1 neuron prediction score. And these are the regions that are accessible in both cases. And to show one example here, this one, it's accessible in Kenyan cells and optic lobe neurons correctly predicted by the model, and the activity shows, yes, Kenyan cells and optic lobe neurons. This time we choose the mutations that doesn't decrease the prediction score for the Kenyan cells, but at the same time decrease the prediction score for the T neurons. And after 9 mutations, we were able to make this enhancer very specific to Kenyan cell. We actually prune the spatial activity of this enhancer. Okay, so far what I showed, everything was by changing single nucleotide at a time, nucleotide by nucleotide approach. Now I will show actually every time we do a mutation, what we saw is either we create or destroy repressor binding sites. And now, okay, instead of these single nucleotides, we can actually implant the transcription factor binding sites if we know what they are. So here the strategy again is quite straightforward. We start from a random sequence and we know which transcription factor binding site we will use. We scan the whole sequence and we calculate prediction score for every position. And the model says this is the best location, and then we implant the motif to that location. And for the Kenyan cells from the previous publication, we knew that ISMB2, 1-cut, and SI transcription factors are very predictive for the enhancer activity. So we use these 4. And to give an example, this initial random sequence and how the nucleotide contribution scores look like. And if we were doing saturation mutagenesis, as you can see, the selected mutations are the ones that destroy repressor binding site. And if we implant the ILIS binding site to every possible location, here you can see these are the locations chosen by the model. And each case, it is again, it prefers the position that it destroys the repressor binding site at the same time while implanting the ILIS binding site. So if we didn't know the repressor binding sites or the code of repressors, imagine we implant the ILIS binding site here, it's in between 2 repressor binding sites, it was going to be non-functional. So thanks to the model, we know where to implant them and we implant them there. And here you see the prediction score from the random sequence, and adding each transcription factor individually. Actually, ILIS and MEF2 were the most effective. Adding ILIS after MEF2 or MEF2 after ILIS had the same effect, and actually it was greater than the sum of their individual contribution. And adding one cut and SR further increased the prediction score, where they didn't contribute much by themselves in the beginning. And we were at the same level with the in silico evolution, sequence evolution, with the 15 after 15 mutation, we are at the same level, but this time it's just 4 steps. And now, like actually the model knows where we implant transcription factor binding sites, and it's the most preferred by the model. Then we can check if there's any preferred distance between different transcription factor binding sites. Here you can see the location of one cut relative to MEF2 and other transcription factors. And as I was saying before, they like to be in the 200 base pair space. But there were two very interesting distance preference for the MEF2 relative to I, one cut and I. MEF2 preferred to be 5 base pairs upstream of the ILIS, where one cut preferred to 3 base pairs downstream of the ILIS binding site. And to see how it looks like and to see to test if it is correct, here you can see this is the initial random sequence. Again, by chance, there are multiple repressor binding sites, and the initial ILIS binding site replaced the repressor, then MEF2 and one cut full of the ILIS binding site. And if we test this 500 base pair in vivo, it was active in the canyon cells as planned. It was a successful experiment. But the only thing that we changed on the random sequence was just adding these 3 binding sites. Then we thought, okay, this is the main driver of the enhancer expression. So we just cut this part, this 49 base pair, and then we tested in vitro. And again, it was very clear canyon cell expression. So we were able to create minimal enhancers that are as functional as their longer version. And this time it's just 49 base pair. And again, I would like to introduce the repressors here as I was doing in melanoma. Okay, from random sequence, we create an enhancer. And again, we choose mutations without touching the activator binding sites we implanted. We choose single mutations that creates repressor binding sites that we don't know which factor it is. It's GATSD most of the time. And when we introduce these mutations and with these 5 mutations and test the activity in vivo, the activity of the enhancer was lost. Again, these repressors, since there are many, they're overcoming the activators and they repress the activity of the enhancer. The last part is about using the gun to generate synthetic enhancers. Since I don't have much time, I am not going to go into detail, but if you have any questions, just find me afterwards. We can discuss what we did by using the guns to generate synthetic enhancers. So to sum up, by using these different strategies, we were able to generate synthetic enhancers from scratch. We were able to increase their activity or decrease their activity level. We were able to rescue them. We were able to augment or prune their spatial activity. We with the help of motif implanting technique, we were even able to make them shorter. Thanks for your attention. Thanks for listening. For the acknowledgement part, my PI Stan Arts, and for different papers, I collaborated with different people. Like for the cross species, it was mainly with Lisbeth. For personal cancer genomes, with Zeynep. Flybrain, we worked together with Jasper and Sarah. And I would like to thank all the members of the Stan Arts lab. I couldn't do any of these without them, actually. And final notice, I actually graduated last week, as I said before, and I'm currently in the job market. If you like to discuss what are the possibilities, please find me. And thank you.